Hello, my dear students. Welcome you all to Sri Jaindra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to start with a very important chapter of human physiology, that is excretory system and osmoregulation. Under this chapter, we will be covering about different types of excretory products and their modes of elimination, osmoregulation, different types of osmolarities. We will also discuss in detail about the excretory system of humans. So heading towards the very first slide, that is introduction. The introduction, excretion, it is a physiologic process. It is a physiologic process in which removal of nitrogenous waste produced during different metabolic reactions, especially the metabolism of proteins and nucleic acids and nucleic acids. The excretion is a physiological process which basically allows the removal of waste, most importantly the nitrogenous waste which are produced during metabolism of proteins and nucleic acids. What is osmoregulation? It is a process of maintenance of osmolarity of the extracellular fluid of the extracellular fluid. This is osmoregulation. So osmoregulation is the maintenance of osmolarity of the extracellular fluid. Then what is osmolarity? How can we define osmolarity? Okay. It is the amount of solutes dissolved per liter of solution. The unit is milliosmoles per liter. Okay. So osmoregulation is the maintenance of osmolarity of extracellular fluid. And what is osmolarity? Is the amount of solutes dissolved per liter of solution. The unit is milliosmol per liter. Okay. So we will be dealing with osmoregulation in detail in the next coming videos. Okay. First, we will be talking about the evolution of different excretory organs of different organisms. So starting from protozoans, in protozoans like amoeba and paramecium, carbon dioxide and ammonia, they are the most important excretory products. They are mostly excreted by diffusion through general body surface. It is considered that the contractile vacuoles also play some role in the removal of excretory product. So very importantly, in case of protozoans, single-celled organisms like amoeba and paramecium, carbon dioxide and ammonia, they are the most important excretory products which are excreted by diffusion through general body surface. So most of the diffusion happens through general body surface, and contractile vacuole secondarily, or it is a accessory excretory organ in case of protozoans, which plays some role in the removal of excretory products. In sponges, when we talk about sponges, in sponges the nitrogenous metabolic waste is ammonia. In sponges, the nitrogenous metabolic waste is ammonia, which leaves the body in the outgoing water current by diffusion. So in sponges, the nitrogenous metabolic waste is ammonia, which goes out through the body in outgoing water current by diffusion. Okay. Most of the sponges are marine and have no problem of surplus water in their cells. Most of the sponges are marine and they don't have any problem of surplus water in their cells. A few sponges like hypotonic fresh water. There are some sponges which lie in hypotonic fresh water and have contractile vacuoles in most of their cells. So most of the sponges, they are marine, so they don't have any problem if they are having surplus amount of water in their cells. But there are some sponges which lie in hypotonic fresh water and they have contractile vacuoles in most of their cells. Flame cells, in case of the flame cell of nephridia, protonephridia, flame cell of protonephridia in case of flatworms, and contractile vacuole in sponges. Okay, so you can see in planaria and fascicola, in planaria and fascicola, that is flatworms, flame cells are here. Okay, also known as protonephridia. Okay, these are contractile vacuoles in sponges. Okay. In coelentrates like Hydra, they also lack special excretory organs. The nitrogenous waste product is ammonia, which is removed through the general surface of the body by diffusion. Some nitrogenous waste products are also thrown along with indigestible matter through the mouth. So in coelentrates, also ammonia is the excretory product. Ammonia can be excreted by only aquatic animals as there is plenty of available water. So ammonia needs large amount of water for its excretion. Okay. Platyhelminthes like planaria, liver fluke, and tapeworm possesses a large number of excretory cells called flame cells or solenocytes or protonephridia, and long excretory ducts, also known as canals of vessels. The flame cells open into the ductules, which in turn open into the excretory duct or canals of vessels. So platyhelminthes like planaria, liver fluke, and tapeworm, they possess a large number of excretory cells called the flame cells or solenocytes or protonephridia, and large or long excretory ducts known as canals of vessels. The flame cells open into the ductules, which in turn open into the excretory duct. Nematoda roundworms, the roundworms such as Ascaris, have H-shaped excretory organ. They have H-shaped excretory organ. It is made up of single rennet cell. So this H-shaped excretory organ is known as single rennet cell in the entire length of the body. It consists of two longitudinal excretory canals connected anteriorly by a network of transverse canals. So this H-shaped rennet cell is basically consisting of two longitudinal excretory canals which are connected anteriorly by a network of transverse canals. A short terminal duct opens outside via excretory pore. Ascaris, it excretes both ammonia and urea. So Ascaris, it excretes both ammonia and urea. So ammonia is highly toxic and highly soluble in water. Thus, it requires large quantities of water for its excretion. Ammonia is the major excretory product in cnidarians, in protozoans, in sponges, flatworms, polychaetes, crustaceans, bony fishes, tadpoles, aquatic amphibians. So ammonia is the major excretory product in all these organisms. Okay. Now comes, so here come nematoda is having H-shaped rennet cell for its excretion. Annelids, earthworm. In earthworm, the excretory system, also known as nephridial system, which consists of three types of nephridia: pharyngeal nephridia, septal nephridia, and integumentary nephridia. So earthworm is having three types of nephridia or nephridial system: pharyngeal nephridia, septal nephridia, and integumentary nephridia. All nephridia commonly called micronephridia. Earthworm excrete 40% urea, 20% ammonia, and 40% amino acids. So earthworm excrete 40% urea, 40% amino acids, and 20% ammonia. Earthworm is mainly ureotelic. Okay, it excretes urea. Chloragon cells found in coelomic fluid also excretory in nature. In case of earthworm, the chloragen cells, which are found in the coelomic fluid, they also serve as excretory function. Blood gland in earthworm found in 4, 5, and 6 segment serves for excretion, manufacturing of blood corpuscles and hemoglobin. So the blood gland in segment number 4, 5, 6 serves for excretion, and also it helps in the manufacturing of red blood corpuscles and hemoglobin. That is why it is known as what? Blood gland. Chloragen cells found in coelomic fluid also are excretory in nature. Earthworm excrete urea, that is why it is known as what? Ureotelic. It consists of three types of nephridia: pharyngeal, septal, and integumentary. All these nephridia are known as what? Micronephridia. This here picture you can see various types of nephridia and their arrangement in Pheretima posthuma, that is earthworm. Okay, pharyngeal nephridia, blood gland. Okay, integumentary nephridia and septal nephridia. This is a rennet cell. These are the vertical excretory ducts, and this is the anteriorly transverse canal. This is the anteriorly transverse canals. Okay. Arthropods, the excretory system of adult prawn, that is crustaceans, consisting of a pair of antennary glands or green glands, a pair of lateral ducts, and a single renal sac. So when we talk about the arthropods, the excretory system of adult prawn, especially the crustaceans, they have a pair of antennary glands or green glands, a pair of lateral duct, and a single renal sac. Insects, centipedes, millipedes, and arachnids like scorpion and spider possesses malpighian tubules as their principal excretory organs. So please remember, in orthopodans, the crustaceans, especially the adult prawn, is having antennary glands or green glands, okay, a pair of lateral duct, and a single renal sac. While when we talk about the insects, centipedes, millipedes, and arachnids like scorpion and spider, they have malpighian tubules as their principal excretory organs. In the malpighian tubules, bicarbonates of potassium and sodium, okay, along with water, uric acid are formed. Large amount of water and bicarbonates of potassium and sodium are reabsorbed by the cells of malpighian tubules and then transferred to the blood, that is hemolymph. Uric acid is carried to the alimentary canal. Uric acid is carried to the alimentary canal of the insect and is finally passed out through anus. So, in case of arthropods, we have two types of excretory systems. One is the green gland or the antennary glands of crustaceans, especially the adult prawn, while in arachnids like scorpion and spider, millipedes, centipedes, and insects, they have malpighian tubules, which act as a principal excretory organ. The Malpighian tubules, they contain bicarbonates of sodium and potassium along with water and uric acid. Okay. Large amount of water and bicarbonates of potassium and sodium are reabsorbed by the cells of Malpighian tubules and then transferred to the blood. Hemolymph. Uric acid is carried to the alimentary canal of the insect and is finally passed through the anus. The antennary gland of prawns. These are the antennary gland of prawns. Okay. Spiders and scorpions possesses Malpighian tubules and coxal glands for both for excretion. So spiders and scorpions, they have Malpighian tubules as well as coxal glands both for excretion. Okay. While the prawns or the adult prawn or the crustaceans, they have a pair of antennary glands or green glands. Now, mollusk. Mollusk, they have organ of Busanus. They have one or two pairs of kidneys or organs of Busanus, also known as Cabot's organ for excretion. So mollusk, they have one or two pairs of kidneys or organs of Busanus and Cabot's organ for excretion. Okay. Echinoderms, they have specialized excretory organs. When specialized excretory organs are absent in echinoderms, example starfish, okay, the excretory products chiefly ammonia, urea, and creatinine are eliminated by diffusion through dermal branchiae or primitive gills and tube feet system. Amoeboid coelomocytes also are excretory in nature. So in case of echinoderms, they don't have specialized excretory organs. For example, starfish, the excretory products chiefly ammonia, urea, and creatinine are eliminated by diffusion through dermal branchiae, that is primitive gills and tube feet. Okay, amoeboid coelomocytes also act as excretory in function. Okay. So in case of invertebrates like protozoa, the contractile vacuole, okay, the excretory waste is ammonia. They are aminotelic, or you know, they also perform osmoregulatory function. Okay, amoeba is ammonotelic while paramecium is osmoregulatory in function. Okay, porifera general body surface function is ammonotelic. It also excrete ammonia. Example, cycon and leucon. Coelenterate ammonia through general body surface, ammonotelic hydra. Platyhelminths, mantis, they have flame cells, solenocytes from the protonephridial system. They are also monotelic, tinea, fascicola, planaria. Nematoda, they have H-shaped rennet cell. Okay, ammonotelic. Example, Ascarius. Annelida, they have nephridial system, three nephridial systems. Okay, they are also monotelic and example Ferretima. When we talk about the earthworm, as I have explained in the previous slide, the earthworm is mainly ureotelic. So please remember, okay, this term. And here they have mentioned that ammonia is, earthworm is ammonotelic. No, it is talking about the annelidans. When we talk about the Ferretima posthuma, that is earthworm, it is ureotelic. Okay. Orthopodans, class Insecta, they have Malpighian tubules, nephrocytes, uricose glands which secrete uric acid, uricotelic, example Praliplata americana, cockroach, housefly, and mosquito. So class Insecta, they have Malpighian tubules or nephrocytes or uricose glands, okay, which forms uric acid. They are uricotelic as they excrete uric acid, example Periplata americana, housefly, and mosquito. Crustaceans, they have antennary or green glands. They have hepatopancreas. They also excrete uric acid, uricotelic, example Peleman. Arachnida, they have coxal glands, Malpighian tubules, hepatopancreas, and nephrocytes. The functions are uricotelic, guanine, and xanthine in small amount. It also excretes uric acid along with guanine and xanthine amino acids in small amount. So please remember, when we talk about that guanine and xanthine mean these are amino acids. So amino acids also act as a excretory product for many organisms. Okay, example spider and scorpion. So insecta they have malpighian tubules, crustacea they have antennary glands or green glands, and arachnida they have coxal glands. Okay, along with malpighian tubules. Mollusca they have organ of Bosarus which act as a kidney, excrete guanine, example unio. Caber's organ also excrete guanine, unio. Renal organs they are ammoniotelic in aquatic and ureotelic in terrestrial. Example Pila and Limex. Renal sacs ammonia in aquatic condition excrete uric acid in terrestrial condition. Okay, mainly excrete guanine as a main amino acid based product. They are ammoniotelic, excrete ammonia and ureotelic as well. Okay. So mollusks they have kidney or organs of Bosanus, which is unio. The excretory product is guanine. Caber's organ excretory product is guanine. Example unio. Renal organs they are monotelic in aquatic and uricotelic in terrestrial regions. Okay, Pila and Limex are the examples. Okay. Echinodermata, they have dermal branchiae, primitive gills, tube feed system, body surface, secretes ammonia and coelomocytes. They are monotelic mainly, excrete ammonia. Example, Cucumaria and Asteris. Okay. Hemichordata, glomerulus or proboscis gland. They are also monotelic. Example, melanoglossus and saccoglossus. Urochordata, they have neural gland and nephrocyte. They excrete xanthine plus uric acid. Example, herdmania. Okay, example is herdmania. Okay, urochordata they excrete, they have neural gland and nephrocytes and they excrete xanthine along with uric acid. Cephalochordata they have distinct categories: protonephridia, solenocytes, brown funnel, renal papillae, Hetzek nephridia. All are ammonotelic. Okay, Amphioxus. Example is Amphioxus branchiostoma. So this is a summary of all different types of excretory organs which are evolved in different organisms for different reasons. Okay, mainly mollusk, annelids, flatworms. Okay, they are important. Excretory products in different organisms. Waste products of protein metabolism and waste products of nucleic acid metabolism. We have actually divided into these two categories. Amino acids. They are the end products of protein digestion absorbed in the blood from small intestine. Certain invertebrates like some mollusks, Unio, Limaceae, and some echinoderms like Asteris excrete excess of amino acids as such. This is called aminotelic excretion or aminotelism. So excretion of ammonia as a main excretory product because of that it gets accumulated because of the end products of protein digestion absorbed in blood from small intestine. Certain invertebrates like some mollusk, Unio, Lemene, okay, and some echinoderms like Asteris, they excrete excess of amino acids as such. They are called aminotelic organisms, okay. Ammonia. In most animals, excess of amino acids are deaminated. So deaminated is basically removal of NH2 group, amino group. That is degraded into their keto and ammonia groups. Okay. So in most of the animals, especially when we talk about the amino acids, they are deaminated, removal of NH2 group, and they are degraded into their keto forms. The keto groups are used in the catabolism for the production of ATP. Okay, the keto groups are used in the metabolism for the production of ATP, adenosine triphosphate, whereas ammonia is excreted as such in the other forms. The ammonia is highly toxic. It is highly toxic and highly soluble in water. Its excretion as such therefore requires a large amount of water. That is why most of the aquatic arthropods, bony and freshwater fishes, amphibian tadpoles, turtles, etc. excrete ammonia. Okay. So these aquatic organisms, they don't have a cushion to store water. They have more amount of water. That is why they excrete ammonia easily. So ammonia is highly toxic and highly soluble in water. Its excretion as such therefore requires a large amount of water. That is why most of the aquatic arthropods, bony fishes, freshwater fishes, amphibian tadpoles, turtles excrete ammonia. This type of excretion is called ammonotelism or ammonotelic excretion. Okay. Ureotelic. The excretory product is urea. It is less toxic and less soluble in water. It is less toxic and less soluble in water than ammonia. Hence, it can stay for some time in body. Most terrestrial vertebrates like adult amphibians, mammals, and such aquatic animals which cannot afford to lose much water. Example, elasmobranch fishes. Elasmobranch fishes. Okay, means marine bony fishes. Elasmobranch fishes. Okay, that is marine bony fishes and elasmobranchs. Adult frog, earthworms, nematodes turn their ammonia into urea for excretion. This type of excretion is called ureotic excretion or ureotelism. So adult amphibians, mammals, okay, cartilaginous marine fishes, marine bony fishes, they all excrete urea. Adult frog, earthworms, nematodes, they turn their ammonia into urea for excretion. Okay. Ammonia is converted to urea in liver by utilizing energy. Ammonia is converted into urea in liver, okay, by utilizing energy. Urea is less toxic and less soluble in water. Hence, there is no requirement of giving out water more to get rid of this waste. Okay. Urea cycle of Krebs Henslate cycle. Urea cycle of Krebs Henslate cycle is basically for the removal of both CO2 and amino group. Urea is formed by the hydrolysis of arginine. By hydrolysis of arginine. So urea cycle of Krebs-Henslate cycle, it is for the removal of CO2 and amino group. Urea is formed by the hydrolysis of arginine. So this process takes place in liver. How? In liver, urea, the chemical formula of urea is NH2CONH2. So the first step you can see, the first step, in this step, the ammonia combines with CO2 along with water, okay? In the presence of carbamoyl phosphate synthetase, carbamoyl phosphate synthetase enzyme, okay, to convert ammonia along with water and CO2 in the presence of carbamoyl phosphate synthetase enzyme, okay, and N-acetyl glutamate, N-acetyl glutamate, okay, is converted into carbamoyl phosphate, phosphate. Okay, so the reaction I will write in the next page. In the mitochondria of liver, this is cytoplasm of liver cells. Okay, so here glutamate basically gives off alpha ketogluteric acid. Okay, now after releasing alpha ketogluteric acid, the glutamate enters the hepatocytes mitochondria. Okay, after converting glutamate into alpha ketogluteric acid. In the first step, what happens? NH2 plus CO2 in the presence of 2 ATP to form 2 ADP to form carbamoyl phosphate. Okay, this is the first step in which NH2 combines with CO2 in the presence of ATP to form ADP to form carbamoyl phosphate. In the second step, the carbamoyl phosphate helps to convert ornithine into citrulline. Okay, now this citrulline, it enters the hepatocyte cytoplasm. It comes outside the mitochondria. Okay, to form arginosuccinate. Glutamate plus aspartate, ATP to inorganic phosphate. Okay, form arginosuccinate, which further forms arginine in the form of fumaric acid. This arginine is converted to ornithine again by giving water okay, so this arginine is converted into ornithine and the cycle gets repeated. There are many enzymes which are required during this process. Okay, so I will write down the process like this is the first, this is the second, then third, then fourth, then fifth. Okay, the enzymes which are required are as follows. For catalyzing the first step, the enzyme is carbamoyl phosphate synthetase. Ornithine transcarbamylase. For second step, ornithine transcarbamylase. Okay, ornithine transcarbamylase. The third step is carried by arginosuccinate synthetase. Okay. Fourth step is arginosuccinate lyase. And the fifth one is arginase. So please remember this Krebs-Henslate cycle. Okay. 3 ATP are used for the formation of one molecule of urea. So according to the energy currency, are used for formation of one molecule of urea. Okay. 4 phosphate bonds are broken for formation of one molecule of urea. Okay. So in the presence of aspartic acid, CO2 and ammonia, urea is formed in liver. Uricotelic organisms, or the organisms which excrete uric acid, it is most expensive excretory product in terms of energy because it requires large amount of energy for its formation. Benzoic acid is a product of fat metabolism. Animals living in dry or arid conditions such as land, gastropods, most insects, land reptiles, snakes and lizards, birds and kangaroo rat, mammal, have to conserve water in their bodies. These therefore synthesize crystals of uric acid from their ammonia for the formation of uric acid xanthine oxidase enzyme. So animals which are living in dry or arid regions such as land, gastropods, most insects, land reptiles, birds, kangaroo rat, they have to conserve water in their bodies. These therefore synthesize crystals of uric acid from their ammonia for the formation of uric acid xanthine oxidase enzyme. For the formation of uric acid, xanthine oxidase enzyme is important. Xanthine oxidase enzyme. Uric acid crystals are non-toxic and almost insoluble in water, hence these can be retained in the body for a considerable time before being discharged from the body. Uric acid is the main nitrogenous excretory product discharged in the solid form. This excretion is called uricotilic or uricotilism. Okay, so excretion of uric acid is known as what? Uricotilism. It is basically conveyed by the animals which live in dry and arid regions such as land, gastropods, okay, land gastropods, land reptiles, snakes, lizards, birds, and kangaroo rat, okay, etc. They have to conserve water in their bodies. These therefore synthesize crystals of uric acid for their ammonia for the formation of uric acid. Okay, so for the formation of uric acid, xanthine oxidase enzyme is necessary. Uric crystals are non-toxic and almost insoluble in water, and then that is why they can be easily retained in the body for a particular time before being discharged from the trimethylamine oxide TMAO. Okay, certain marine mollusks, crustaceans, and teleosteik fishes first form trimethylamine from their ammonia by a process known as methylation. Then the trimethylamine is oxidized to trimethylamine oxide for excretion. This oxide is soluble in water but not toxic. So here ammonia is converted into TMAO. It is soluble in water but non-toxic. Some telostick, some telosticks fishes excrete it, but elasmobranchs retain it. Elasmobranch fishes retain it. Telostick fishes, they excrete it. Retain it. Okay. In mammals, a small amount of uric acid is produced by the metabolism of purines. Okay. Guanine. Spiders typically excrete their ammonia in the form of guanine. Some guanine is also found in unio, penguin, birds, and it is insoluble in water. Hence, no water is required for its excretion. So, guanine comes from the purine metabolism. Okay, it contains phosphorus. It contains phosphorus. It is insoluble in water and excreted in the form of crystals in spiders, and some birds are the examples of guanine. Spiders and some birds. Now comes the waste products of nucleic acid metabolism. As a result of nucleic acid digestion, nitrogenous organic bases like purines, adenine, and guanine, and pyrimidines, cytosine, thymine, and uracil are absorbed from the intestine into the blood. Most of these are excreted out. About 5% of the total excretion of body accounts for these substances. In man, purines are changed into uric acid for excretion. Very important. In most other mammals, nitrogenous organic bases are excreted in the form of allantoin. Okay, in most other mammals, the nitrogenous organic bases are excreted in the form of allantoin. Insects, amphibians, reptiles, and birds also excrete these bases in the form of uric acid. Some freshwater mollusks and crustaceans arthropods excrete these in the form of ammonia. The freshwater mollusk and crustaceans arthropods, they excrete in the form of ammonia. There are some excretory substances which are sun-dry or other excretory products like hippuric acid or ornithuric acid. The mammals excrete benzoic acid as hippuric acid. Mammals excrete benzoic acid as hippuric acid. Okay. Sometimes food of rabbit and other mammals may contain traces of benzoic acid, or this acid may be formed in small amounts during fat metabolism. It is highly toxic, it is absorbed in blood, it it combines with glycine and changed into less toxic hippuric acid. So benzoic acid is converted into hippuric acid. Okay. In birds, benzoic acid is combined with the ornithine and changed into ornithuric acid for excretion. So hippuric acid, the benzoic acid is converted into the hippuric acid. Okay, because benzoic acid combines with glycine to form less toxic hippuric acid. Benzoic acid is highly toxic. Okay, in birds, benzoic acid combines with ornithine and change into ornithuric acid. Okay, creatine and creatinine. Muscle cells contain molecules of creatine phosphate, which are high energy molecules and serve for storage of bioenergy like ATP. It is synthesized by three amino acids: glycine, arginine, and methionine. Excess amount of this phosphate is, however, excreted out, such as or after being changed into creatine. So creatine is basically converted into creatinine phosphate or creatinine, okay, which is removed from the body. This is creatine, which is formed in liver by the metabolism of glycine, arginine, and methionine. At the condition of rest, it is converted into creatine phosphate, which is stored in muscles and can be converted into creatine waste. Okay, so creatine phosphate converted into creatine during exercise. Okay, now we are going to start with excretory system of men. The excretory system of men, I will be starting in the next turn. Till then, bye bye, take care, stay safe, stay healthy. Please go through my previous videos. I will be starting the excretory system of men in the next turn. Bye bye, take care.